Welcome to CBSE IB Academy. My name is Lutfi Hawk. Today, we're going to be talking about the fundamental unit of life in one shot. If you're interested in attending our weekly live classes where we cover additional questions on every topic and have a live Q&A and cover the full chapter on the fundamental unit of life, there will be further details at the end of the video and in the description box below. So let's go ahead and get started with the first topic, which is the cell. So before Robert Hooke, microscopes were first invented in 1590. That was microscopes. Then Robert Hooke, in the year 1665, that's when he coined the term cell. And that was after he observed dead cork cells under a microscope, and he saw these honeycomb-like cavities. And from looking at that, he coined the term cell. Now, it's also important to keep in mind that in 1674, a gentleman by the name of Antoine van Leeuwenhoek, which is spelt like this, he coined the term animalcules. And that was after he observed live cells under a microscope. So microscopes were invented in 1590. The first dead cells were observed by Robert Hooke in 1665, and he coined the term cell. And then roughly 10 years later, Antoine van Leeuwenhoek, in 1674, coined the term animalcules after observing cells, live cells, under a microscope. So you should keep those 3 dates in mind, or those 3 years. So now let's take a look at cell structure and organization. So the fundamental unit of life is the cell in biology, almost like how in chemistry the fundamental unit is the molecule, and in physics, the atom. In biology, it's considered to be the cell. So if we take the stomach, for example, you'll have different cell types. You might have mucus cells, which produce mucus. You might have parietal cells, which produce acid. You might have chief cells that produce pepsinogen and lipase that digest protein and lipids. And the point is that with these different cell types in the stomach, you will form a particular tissue. Now, you can have a variety of different tissue types. You can have an epithelial tissue. You might have muscle tissue, all in the stomach, for example. You may have nervous tissue. You may have connective tissue. And all these tissue types will give rise to an organ. And in this case, we're discussing the stomach. Now, what's really beautiful is that you can have a whole variety of different organs. The stomach, the liver, the small intestine, the large intestine, gallbladder, and the list can go on and on. And these organs can interact with each other and give rise to a single organ system. And in this case, it would be the digestive system. And the human body, I'd say, has roughly 11 organ systems. The digestive system is just one. You'll also have the muscular system. You'll have the lymphatic system, nervous system, and on and on and on. And all these different organ systems come together to give you the organism. And in this case, we're discussing the human. So that's a little bit about cell structure and organization. Cell theory. There are 3 individuals that you should know when it comes to cell theory. The first is Matthias Jacob Schleiden, In the year 1838, he claimed that all plants are made from cells. Next, you have Theodor Schwann, who in 1839 claimed that all animals are made from cells. And then lastly, you have Rudolf Virchow, who in 1855, he really popularized the statement omnis cellula e cellula, which means that every cell is derived from a pre-existing cell. It's just important to keep in mind that while he popularized that statement in the context of cellular pathology, it was actually first said and stated by Francois Vincent Respel. It was first coined by him in 1825, but it was actually Rudolf Virchow who popularized that statement that all cells come from pre-existing cells later in 1855. All right, so I'll clear the ink and ask you your first three questions. Schleiden claimed what and in what year? He claimed that all plants are made from cells in 1838. Schwann claimed what and in what year? That all animals are made from cells, and that was in 1839, a year later. Virchow popularized what statement in what year? It was the statement omnis cellula e cellula, that every cell is derived from a pre-existing cell, and he popularized that in 1855. Okay, next topic. The prokaryote. So cells can be of two types. They can be prokaryotic or they can be eukaryotic. And now I'm going to give you a little map here. If you're under the domain of bacteria or archaea, then you are a prokaryote. But if you are a protist or fungi or an animal cell or a plant cell, you will be a eukaryote. So you should keep these divisions in mind for prokaryote versus eukaryote. So here are the basic components of the prokaryotic cell. In red, we have the plasma membrane, which encloses the cytoplasm. In teal, we have the cell wall, which is a rigid structure. And in gold, we have the capsule. And the capsule is a jelly-like substance that coats many prokaryotes. And there are these finger-like projections from the capsule called fimbriae. In brown, what do you think these components are? Yep, ribosomes. They're involved in protein synthesis. In purple here, we have the nucleoid, which is the genetic material of the prokaryote. And lastly, in orange, we have the flagella, which is made from flagellin and is involved in cell motility. So those are the basic components of the prokaryote. Let me clear the ink and ask you your three questions. One thing I'll point out is that a core concept for prokaryotic cells is that they do not have true membrane-bound organelles. So for example, the nucleoid is not a true membrane-bound nucleus. It's just where the prokaryotic cell stores its genetic material. Okay. First question, how do prokaryotes generate energy? Glycolysis, mitochondria, ATP synthase, or ANC? And the answer is ANC. That is because prokaryotes do not have mitochondria, that is a membrane-bound organelle. They can undergo glycolysis, which is a metabolic process that occurs in the cytoplasm that produces ATP anaerobically. They do have, or can have, ATP synthase, and it'll be embedded in the infoldings of the plasma membrane, and it'll harness a hydrogen ion potential to produce ATP. Second question, what shape is prokaryotic DNA? It's linear, circular, both, or neither? And the answer is circular. Prokaryotic DNA is a circular chromosome. Are prokaryotes unicellular or multicellular? And the answer is unicellular. Eukaryotes have linear DNA molecules and can be multicellular, but prokaryotes have circular DNA chromosomes and are unicellular. Next topic is the eukaryotic cell. We'll revisit comparing eukaryotic cells and prokaryotic cells at the end of the lecture. But first, let me just guide you through a panoramic view of the eukaryotic cell. First, you have the plasma membrane. The plasma membrane has various functions, and I will go over the main ones now. The first is cell-to-cell recognition. The second is import and export of various nutrients and ions. And the last is you can have extensions of the plasma membrane, for example, pseudopodia, that are involved in motility. Those are three functions of the plasma membrane. Okay, now let me walk you through the other components of the eukaryotic cell. Here in purple, you have the nucleus, which stores the genetic material of the eukaryotic cell. In brown, like before, you have the ribosomes, which are involved in synthesizing proteins. Then in blue, you have the endoplasmic reticulum. The ones studied with ribosomes are the rough ER, while here to the left, the endoplasmic reticulum that's not studied with ribosomes, is the smooth ER. Smooth ER. The rough ER and the smooth ER will be involved in processes like protein modification, calcium storage, and calcium ion storage, and steroid synthesis. We'll go into that in more detail later in the lecture. Next, in green, you have the Golgi, which is like a packaging center for the cell. It'll mark different components for their final destination. It also modifies proteins and lipids. In magenta, you have lysosomes, which are involved in autophagy and digestion. In this beautiful orange color, you have mitochondria. which are the powerhouse of the cell, a site where ATP is produced through oxidative phosphorylation. In pink, you have cytoskeleton elements here. So I'll just say cytoskeleton, and we'll go over the 3 types of cytoskeleton elements later, which is the microtubule, the intermediate filaments, and microfilaments. In black, you have centrioles, which are involved in, which are involved in microtubule formation. And then lastly, in red, you have the flagellum, which is involved in motility. And that is your overview of the eukaryotic cell. Obviously, in white here, you have your cytoplasm as well. Okay, I will clear the ink and then ask you your 3 questions. Which components of the eukaryotic cell are animal cell specific and not present in plant cells? Centrioles, flagella, centrosomes, or all of the above? Which is not present in plant cells? And the answer is all of the above. Plant cells do not have centrioles, centrosomes, or flagella. Which of the following are not found in prokaryotes? Ribosomes, mitochondria, DNA, or flagella? And the answer is mitochondria, because prokaryotes cannot have membrane-bound organelles like the mitochondria, but they do have ribosomes, they do have DNA, circular DNA chromosomes, and they do have flagella. Which eukaryotic cell component is not present in prokaryotes? Ribosomes, DNA, nucleus, or flagella? And the answer is nucleus, because that is a membrane-bound organelle. Instead, prokaryotes store their DNA in the nucleoid. All right, the nucleus. Most of the cell's genetic material is stored in the nucleus, but it's important to keep in mind that some DNA is stored in the mitochondria and chloroplasts. Those organelles will have their own DNA. The nuclear envelope, which is right here, the nuclear envelope, encloses the nucleus and it separates it from the cytoplasm. It's perforated with nuclear pores, and you'll have within those nuclear pores these nuclear pore complexes. So these are your pore complexes. Pore complexes. So your pore complexes are about 100 nanometers in diameter, the open portion, 100 nanometers. But they're selective in what they allow into and out of the nucleus. Now, on the inside of the nuclear envelope, which would be these regions here, you have a cytoskeleton element that forms a cage net-like structure called the nuclear lamina. And this is important. It's called the nuclear lamina. And the nuclear lamina is a cytoskeleton element. It is made from intermediate filaments. And it basically provides structure to the nuclear envelope. The nucleus stores DNA in the form of linear DNA molecules called chromosomes, and when you add packaging proteins to DNA, you call it chromatin. So chromatin is DNA plus protein. In non-dividing cells, you'll have this distinct circular region called the nucleolus. Nucleolus. In the nucleolus is the site of rRNA synthesis, and also the assembly of small and large ribosomal subunits. So that's the nucleolus. Okay. It's important to keep in mind there's a concept called the central dogma that I want to highlight before we move on to the next topic. And the central dogma is it has like three components. It starts with DNA, then it has RNA, then it has protein. And I just wanted to point out that in the central dogma, this first step of DNA to RNA, which is transcription, this occurs in the nucleus. And then the second part of the central dogma, which is the translation of RNA into protein, that will generally occur outside of the nucleus on the rough ER or in the cytoplasm. And we'll discuss that in more detail later. So that is the nucleus, and we will go ahead and clear the ink and ask you your first three questions for this topic. Which of the following belongs to prokaryotes? Nucleolus, nucleoid, nucleus, or A and C? And the answer is nucleoid. That belongs to prokaryotes. The nucleolus and the nucleus are eukaryotic elements. The nuclear lamina is made from what cytoskeleton element? Is it made from microtubules, microfilaments, intermediate filaments, or none of the above? And the answer is intermediate filaments. If you have a cytosolic protein, which is 50 nanometers in size, and it's highly concentrated in the cytosol, so outside of the nucleus, if you allow that cell to rest for 24 hours, would you expect the concentration of that protein to equilibrate between the cytosol and the nucleus, keeping in mind that the diameter of the nuclear pore complex is roughly 100 nanometers in size, so this protein is smaller? And the answer is no. You would not expect it to equilibrate necessarily, and that's because these nuclear pore complexes are selective, and it's not just size selective. They're selective in what kinds of proteins or other molecules they allow to enter or exit the nucleus. On to the next topic. The ribosome. Ribosomes read mRNA transcripts and synthesize proteins via a process called translation. It has a small and large subunit, and each of these subunits is made from multiple proteins. Now, the small subunit has, in addition to the multiple protein subunits, a single rRNA molecule, while the larger subunit has 3 rRNA molecules. And in eukaryotes, the ribosomes are called or denoted ADS ribosomes, while in prokaryotes, it is 70S for prokaryotes. Okay. Keep in mind that the small and large subunits are assembled together in the nucleolus, and that if a cell produces a lot of protein, you'll expect it to have a lot of ribosomes. Okay. Now, ribosomes can come in 2 flavors. They can be either bound or they can be free. If a ribosome is bound to the rough ER, then you expect it to be synthesizing proteins that will be either secreted or embedded in a membrane. If a ribosome is free in the cytosol, you'll expect it to produce, for example, cytosolic proteins. Let's go ahead and clear the ink and ask your first 3 questions for this topic, but before I do that, I wanted to point out here in the top left, in this next to this blue symbol, I've written antibiotics. Whenever we're covering a topic in CBSE biology that I think has a connection to medicine that's important, I'll try to point it out. So because eukaryotes have 80S ribosomes, and they're distinct from the prokaryotic 70S ribosomes in many ways, or in different ways, that distinction allows us to have or make or isolate different classes of antibiotics that are specific for just the prokaryotic ribosome, and allows us to target and kill bacteria only and not eukaryotic cells by targeting just what's specific in a 70S prokaryotic ribosome versus a eukaryotic one. Okay, so I'll go ahead and clear the ink and ask the questions. What is a ribosome made of? Protein, mRNA, rRNA, or A and C? And the answer is A and C. A ribosome is made from protein and rRNA. A pancreatic cell produces a large amount of secreted enzymes. Would you expect there to be a higher ratio of bound or free ribosomes in this pancreatic cell? And the answer is you would expect there to be a large amount of bound ribosomes. And that's because the bound ribosomes will produce can produce secretory proteins. Last question is, are ribosomes found in animal cells, plant cells, prokaryotic cells, or all of the above? And the answer is all of the above. Ribosomes are found in animal cells, plant cells, prokaryotic cells. They're found in the nucleus, and they are also found in, they're assembled within the nucleus, and they are embedded also in the nuclear envelope. And I believe you can also find them in mitochondria and chloroplasts. Okay. The endoplasmic reticulum. The endoplasmic reticulum is an extension of the nuclear envelope. The ER has 2 distinct but connected regions. You have the rough ER, which is studded with these ribosomes, and then you have the smooth ER, which is not studded with ribosomes. So let's start off with the rough ER first. So the rough ER. It's involved in producing secretory and membrane proteins. So secretory and membrane proteins. And it's also involved in protein modification. So it's not just making these secretory membrane proteins, but it's also modifying them. Okay. When a secretory membrane protein is being translated on an ER-bound ribosome, that protein gets threaded into the ER, and then it can be further modified in the ER, and that kind of modification is called glycosylation. Glycosylation. That's when a sugar will be added to the protein. The protein then can continue on to the Golgi and then to its final destination. Another function of the rough ER, besides making secretory and membrane proteins and modifying them, is actually membrane synthesis. So the ER will grow in size. It'll add phospholipids and proteins to itself, and then it'll section itself off and send it off to different parts of the cell, for example, the plasma membrane in the form of vesicles. So it's involved in membrane synthesis. So there you go. Those are 3 functions of the rough ER. And now let's look at the smooth ER. Smooth ER. So what are the functions of the smooth ER? It's involved in lipid synthesis. It'll make, for example, steroids, the female and male sex hormones. It's also involved in carbohydrate metabolism. For example, the last step of gluconeogenesis, which is the process of producing glucose, occurs in the smooth ER. That's when glucose is converted, it's when glucose 6-phosphate gets converted into glucose, and that occurs in the smooth ER. And the third function is drug and poison detoxification. The smooth ER is involved in that. That means that, for example, a hepatocyte, which is a liver cell, has enzymes that will add hydroxyl groups to alcohol. So say you have a drug or a poison, and the hepatocyte will add these hydroxyl groups to, for example, alcohol or barbiturates or other drugs, and help flush them out of your system. Okay, and the fourth function of the smooth ER is calcium ion storage. In muscle cells, the smooth ER, for example, keeps a large calcium store. And then when it's activated, it releases that calcium, and that leads to muscle contraction. So those are the 4 functions of the smooth ER. 1, 2, 3, and 4. I'll go ahead and clear the ink and ask you your questions for this topic. First question. A protein is being translated on a rough ER ribosome. It's cleaved off, and that protein ends up in the ER lumen. What are the possible destinations for this protein? Can it end up in a lysosome? Might it be secreted? Might it end up in the plasma membrane? Or maybe A and B? And the answer is A and B. If a protein's cleaved off into the ER lumen, then it'll be either secreted, or it'll maybe end up in the lumen of a lysosome. But it won't be membrane bound. It won't be like embedded in a membrane. For that to be the case, it would have to remain embedded in the ER membrane. It can't be cleaved off into the ER lumen. So not the plasma membrane. Second question. A protein is being translated on a rough ER ribosome and remains embedded now in the ER membrane. What are some possible destinations for that protein? Plasma membrane, ER membrane, Golgi membrane, or all of the above? And the answer is all of the above, because it's still embedded in the rough ER membrane. Third question, comparing a pancreatic cell versus a male testes cell. Which will be more rich in smooth ER, and which will be more rich in rough ER? And the answer is the pancreatic cell, which is producing a lot of secretory proteins. You'd expect it to be rich in rough ER, because that's the ribosomes in the rough ER help produce secretory proteins. And then the male testes cell, which produces the male sex hormone testosterone, will be rich in smooth ER. And that's because the smooth ER is involved in steroid synthesis. There you go. Next topic. The Golgi. So the Golgi is involved in receiving, sorting, and shipping cell products from the ER to the rest of the cell. The Golgi is composed of these stacked membranes called cisternae. And the Golgi stack has directionality. That means that vesicles enter on the cis end and they exit at the trans phase. So according to the cisternal maturation model, cisternae move and mature from cis to trans. Now some vesicles will move forward in the Golgi, like here, but some will also move backwards in the Golgi, and they can even go backwards from the Golgi to the ER. And the Golgi is also involved in modifications. So, excuse me, modifications. And I'll go over those modifications that the Golgi can do. First is glycosylation. That's when you add a sugar to a protein to modify it. It can also do something called sulfation. That's when a sulfur can be added to the protein. And the last is phosphorylation. And that's when a phosphate group can be added to the protein. So those are three methods of modification that the Golgi can undergo on proteins. Let me go ahead and clear the ink and ask you your questions. First question, who discovered the Golgi and in what year? You do have to know this. And the answer is Camillo Golgi. And in what year? The year of 1898. 1898. Camillo Golgi discovered the Golgi. The Golgi contents are transported forward and backward via what? Vacuoles, vesicles, both, or none of the above? The answer is vesicles. Compared to an epithelial cell, a pancreatic cell, which produces a large amount of secretory proteins, will be rich in which of the following? Rough ER, Golgi, ribosomes, or all of the above? And the answer is all of the above. You will have more ribosomes, and they'll be embedded on more rough ER, and you will have more Golgi as well. And that will all help this pancreatic cell produce a lot more secretory protein than, say, an epithelial cell. Next topic is the lysosome. So the lysosome is a membranous sac of hydrolytic enzymes that many eukaryotic cells use to carry out either intracellular digestion or a process called autophagy. So in intracellular digestion, a cell phagocytoses food into a vacuole, a food vacuole. Okay? And then that food vacuole will go ahead and merge with a lysosome, which has these hydrolytic enzymes inside of it. And then those enzymes will go ahead and degrade the food. And that is the process of digestion. In autophagy, a damaged organelle, or a part of damaged organelle, for example here maybe a part of the mitochondria, will be enclosed in a vesicle. And then that will go ahead and fuse with the lysosome. And then the hydrolytic enzymes will go ahead and digest and recycle the components of that damaged part of the organelle. Now, it's important to keep in mind that enzymes within the lysosome, they function at a low pH. So now that means that in the low pH environment of the lysosome, so that means that there's a high proton concentration, these enzymes will function, okay? And they will degrade proteins, for example. But if a lysosome ruptures for whatever reason, right, and these come out into the cytoplasm, if the pH is neutral, say about 7, which the cytoplasm is, it's roughly a neutral pH environment, then these hydrolytic enzymes generally will not function. So they won't wreak havoc on the cell. if, for example, a cytoplasm ruptures. If a lysosome ruptures and the enzymes come out. They function at low pH. Okay? Now let me go ahead and clear the ink and ask you your questions. So a liver cell, it's known as a hepatocyte. It recycles half of its cellular components weekly. Would you expect that hepatocyte to be rich or deficient in lysosomes? And the answer is rich. You expect it to be rich in lysosomes because it will help in the process of autophagy, in the process of recycling its cellular components. The medical connection here today for the CBSE topic of lysosomes is Tay-Sachs disease. So in Tay-Sachs disease, there's a lysosomal protein, hexosaminidase A, which is mutated. Now, if that lysosomal protein is mutated, what kind of pathophysiology would you expect to see? And the answer is, you would expect to see, without a functional hexosaminidase A protein, what would happen is that cells would be unable to hydrolyze and break down lipids, certain kinds of lipids. And this would lead to lipid accumulation and then subsequent cell death, like nerve cell death in the brain and spinal cord. So that's what can happen in Tay-Sachs disease when you have a mutation in lysosomal protein. Okay, third question. If lysosomes fuse with the outer membrane of a mitochondria in a cell, and say that the components spill into the intermembrane space, would you expect to see any cellular damage? And if so, what cellular process would it affect? So to answer this question correctly, you have to keep in mind that if the lysosome fuses with the outer membrane, the hydrolytic enzymes will enter the intermembrane space. And the intermembrane space of the mitochondria has a low pH because there's lots of hydrogen protons there. So those enzymes would function in that environment. And those hydrolytic enzymes could start degrading proteins, and that would affect ATP production in the mitochondria. Okay, next topic. The vacuole. So vacuoles are large vesicles that are derived from the Golgi and ER. The vacuole's membrane is selective in transporting solutes into and out of itself, which leads to it having a distinct composition compared to the cytoplasm. In unicellular eukaryotes, so in unicellular eukaryotes, you can have something called contractile vacuoles. And those are involved in pumping excess water out of the cell. In plants, you can have a large central vacuole, and they can be involved in storing organic compounds for energy. So you may store in the vacuole compounds for energy, so we'll just say storage, possibly for energy. You can store compounds for pigments that might attract pollinators, or maybe you might be storing poisons, and that might be to protect itself from herbivores. So plants can have vacuoles that store energy, rich molecules, or it can store pigments to attract pollinators, or it can store poison to fend off and protect itself from herbivores. Keep in mind that vacuoles, when they're filled, they give plants they make plant cells turgid, which means that they're nice and like taut. They have structure. This large central vacuole in plant cells can also, one last thing, that they can store the plant cells' storage of potassium and chloride. The contents of a plant's vacuole is often called sap. Okay, so that is the vacuole. Go ahead and clear the ink. One moment. Your first question is, are vacuoles present in both animal and plant cells? If so, in which is it larger? And the answer is yes. You can have vacuoles in both animal and plant cells, but they'll be larger generally in plant cells. A vacuole in a plant cell can occupy up to 90% of the plant cell's volume. Is that true or false? Answer is true. Lastly, what is the name of the membrane that encloses the chief vacuole of a plant cell? That membrane is called a tonoplast. Next topic. The mitochondria. So mitochondria are the sites of cellular respiration. That's a metabolic process where oxygen is used to drive the production of ATP by extracting energy from like sugar, fats, and proteins. So what's really interesting is that cells can have literally hundreds to thousands of mitochondria. So you can have many of them. And the number correlates with the cell's level of metabolic activity. Okay. Now, you can have in the mitochondria, you have two membranes that enclose it, and they're both phospholipid bilayers with a unique collection of proteins. The outer membrane, here in orange, is a smooth membrane. And it has pores that allow the selective entry and exit of proteins. The inner membrane in black, this is convoluted and folded, and that is for a particular reason. The inner membrane has all these infoldings in order to increase surface area. So you can have a lot of ATP synthase molecules embedded in it so that you can produce a lot of ATP. Now between and these infoldings of the plasma membrane, they're called, like here and here and here, the infoldings are called cristae. Okay, so this inner and outer membrane creates a intermembrane region, and that intermembrane space is between the black and orange membranes, and that has a high concentration of hydrogen ions. And then lastly, in this beautiful deep orange color here, you have the matrix. Okay, and that's in solid orange. So the matrix houses mitochondrial DNA, which is here in blue, and it also houses ribosomes. Hydrogen ions, like I was saying before, will concentrate in the intermembrane space, and then they'll flow back down their concentration gradient into the matrix vis-a-vis ATP synthase proteins that are embedded in this inner membrane, and that will produce ATP. And those are the core points about the mitochondria. So here in the top left, whenever I find or come across a topic in CBSE biology that I just think is fascinating, I'll point it out. And in this case, I wanted to draw your attention to the ATP synthase molecule. It's a molecular rotary motor. It's made up of about 20 subunits. And this thing is just fascinating. You know, it can harvest the voltage difference or ion potential across that inner membrane of the mitochondria, and it can fuse ADP and an inorganic phosphate ion in order to produce ATP. And it does this by rotating like a rotary motor, and it's just very neat, and I just wanted to draw your attention to that. Okay, so let's go ahead and clear the ink and ask the questions. Question 1. A detergent perforates the outer membrane of the mitochondria. Would you expect cellular respiration to be disrupted? And the answer is most definitely. You would expect it to be disrupted because the H plus gradient will dissipate and disappear, and you won't be able to produce any ATP without that concentration gradient of H plus. Which of the following processes does not occur in mitochondria? The citric acid cycle, the electron transport chain, oxidative phosphorylation, or the Calvin cycle? And the answer is the Calvin cycle. The citric acid cycle occurs in the matrix. The ETC is embedded in the inner membrane. And oxidative phosphorylation is that process of using oxygen as the final electron acceptor in the electron transport chain to establish an H plus gradient and then translate that energy into fusing ADP and PI into ATP. So answer is Calvin cycle. That's not present in mitochondria. That's in chloroplasts. Which of the following are functions of mitochondria? Fatty acid oxidation, non-shivering thermogenesis, oxidative phosphorylation, or all of the above? The answer is all of the above. The mitochondria will oxidize fatty acids. They can allow H plus ions to flow down their gradient without producing ATP, but instead they just produce heat or thermal energy, and they can undergo and the mitochondria can do oxidative phosphorylation to produce ATP. All right, the next topic. Plastids. So plastids are membrane-bound organelles found in plant cells. They include chloroplasts, which are involved in photosynthesis. But you can also have synthesis. You can also have chromoplasts, which store pigment. Like we were saying earlier, maybe to attract pollinators. And then lastly, you can have leucoplasts. And leucoplasts are non-pigmented, but they can have various functions. They can store like various complex carbohydrates. Okay, so those are three kinds of plastids. Do prokaryotes such as cyanobacteria contain chloroplasts? And the answer is cyanobacteria do not have chloroplasts because that is a membrane-bound organelle. Which plastid is non-pigmented? Chloroplasts, leucoplasts, or chromoplasts? And the answer is leucoplasts. Those are non-pigmented. What are some special types of leucoplasts, and what do they store? So we should go over this. amyloplasts, oleoplasts, or proteinoplasts? And the answer is all of the above. So amyloplasts, they store starch, which is a complex carbohydrate. Oleoplasts, they're involved in lipid synthesis and storage. And proteinoplasts, as you can guess, they're involved in protein storage. So those are three different types of leucoplasts: amyloplasts, oleoplasts, and proteinoplasts. And then you have chromoplasts that store pigment, and then chloroplasts that are involved in photosynthesis. All right, so let's discuss chloroplasts in more detail. So chloroplasts are organelles in plant cells that contain the green pigment chlorophyll. That is the green pigment. They're involved in photosynthetic production of sugars, and the chloroplasts have an inner membrane. Here in like faint gray, they have an inner membrane, but they also have a outer membrane. Okay, and because of the inner and outer membrane, you then have a intermembrane space. There's also another membrane system inside the chloroplasts, and that's in the form of interconnected sacs called thylakoids. Right here, thylakoids. So that's another membrane system within the chloroplast. Now, a stack of thylakoids is called a granum. So one whole stack of thylakoids, you call that a granum. All right, so there are three spaces you should be able to identify in a chloroplast: the intermembrane space, which is between the inner and outer membranes; the stroma, which is right here, the stroma, which has, the ribosomes and the DNA molecules of the chloroplasts. And then you have the thylakoid space, which is inside the thylakoid, right here, the thylakoid space. Thylakoid space, it's just the space inside the thylakoid. So these are three spaces you should know: thylakoid space, intermembrane space, and the stroma. Okay. Now, in photosynthesis, you have light reactions and dark reactions. That's what they're referred to as, light reactions and dark reactions. And what you should know is that the light reactions occur within the thylakoid space, and that the dark reactions occur in, I believe, the stroma. Correct. The dark reactions occur in the stroma, and the dark reactions are also called the Calvin cycle. And those are the core points to know about chloroplasts. In the light reactions, where do protons concentrate, and which way do they flow? So, do they concentrate in the intermembrane space between the inner and outer membranes, like in the mitochondria? Or do they concentrate in the stroma, or do they concentrate in the thylakoid space? And the answer is that they concentrate within the thylakoids, and then they flow down their concentration gradient into the stroma, and they go through ATP synthase molecules and produce ATP. Okay. Question number 2. Where do the dark reactions or the Calvin cycle take place? They take place in what? The intermembrane space, stroma, or thylakoids, or none of the above? The answer is stroma. And lastly, in what space are the chloroplasts, ribosomes, and DNA found? Are they found in intermembrane space, stroma, or thylakoids? And the answer is the DNA and ribosomes are found in the stroma. All right. The cytoskeleton. There are many functions to the cellular cytoskeleton, but let's first look at the 3 main types of fibers that make up the cytoskeleton. That would be the microtubules, the microfilaments, and the intermediate filaments. So now all eukaryotic cells have microtubules, which are hollow rods made from the globular protein tubulin. Now each tubulin protein is a dimer made from alpha tubulin and beta tubulin. So each tubulin protein is made from alpha tubulin and beta tubulin. It's a dimer. Now there's a sense of directionality with microtubules, as in one end, which is called the plus end, there is a rapid assembly and disassembly of tubulin dimers that occur at a much faster rate than the other end, which is the minus end. Microtubules have various functions, and we'll go over that now. Microtubules are involved in the following. Number 1, let me zoom in here. Microtubules are involved in the general support and structure of the cell. Number 2, they serve as tracks along which vesicles and organelles can be transported via mortar proteins, say from the ER to the Golgi to the plasma membrane. And then lastly, in chromosome movement. Microtubules are involved in chromosome movement. When we cover mitosis, say in another video or lecture, you'll see that the microtubules can attach to chromosomes and are involved in moving chromosomes. So these are three functions of microtubules. In animal cells, keep in mind that microtubules grow out of the centrosome, which is a region that is found near the nucleus often. Now, within the centrosome, you'll have a pair of centrioles, which, if you remember, was in the original eukaryotic cell diagram. You should probably write this down. Each centriole is composed of 9 triplets. Yes, 9 triplets of microtubules, and they're arranged in a ring formation. Now, last important thing about microtubules is that plant cells, while they have microtubules, they do not have centrosomes or centrioles. Okay, so centrioles are not in plant cells. Okay, not in plant cells. I think one other thing I'd mention about microtubules is that animal cells can have microtubule containing extensions of the plasma membrane, and that would be your cilia, for example. And a really interesting medical concept is the mucociliary escalator. This is in the trachea of your lungs, and you basically have these ciliated lining within your trachea that sweeps up mucus outside, up and out of your lungs in order to eliminate debris within your lungs. So that's a microtubule mediated process, because those microtubules protrude from the plasma membrane and form these cilia that help sweep up debris and mucus up and out of your lungs. All right, on to the second cytoskeleton element, which is the microfilaments. Microfilaments are these thin solid rods, and they're made from the globular protein actin. A microfilament is a twisted double chain made from these actin subunits, and it's generally linear, but it can bind onto other proteins and then form these branch networks. For example, there's a mesh network of microfilaments just under the plasma membrane, and that's called the cortex. It's just under the plasma membrane. Muscle cells also will have microfilaments that interact with myosin proteins, and it will be involved in muscle contraction. And then lastly, microfilaments can also help form pseudopodia. And so hence it's also involved in motility. There's a lot of information here with the cytoskeleton elements, but I'm just trying to highlight the most important ones. And like microtubules, microfilaments are found in all eukaryotic cells. Okay. Lastly, intermediate filaments. So intermediate filaments are only found in some animal cells. Okay? Animal cells. Intermediate filaments. They are a diverse class of cytoskeleton elements because each type of intermediate filament is constructed from a particular molecular subunit that will belong to a particular family of proteins. For example, maybe from the family of proteins from keratin. Okay. Now the size of these subunits may vary. So microtubules and microfilaments are like consistent in the diameter and composition of their subunits and their elements, but in intermediate filaments, it's more varied. Okay. Also, intermediate filaments are typically more permanent fixtures in the cell than microtubules or microfilaments, which are disassembled and assembled very frequently, but the intermediate filaments are more fixed and permanent fixtures. For example, if you took a cell and you chemically treated it to remove microfilaments and microtubules and the cytoplasm, and what you would see, what would be left of that living cell, would be just a web of these intermediate filaments that help retain the shape and structure of the cell. And if you remember, the nuclear lamina was made from intermediate filaments. All right. That was quite a lot. So what questions should we keep in mind for the cytoskeleton? Are centrioles present in plant cells? Answer is no. What cytoskeleton element are centrioles made from? Answer is microtubules. And is that cytoskeleton element present in plant cells? The answer is yes. So no centrioles in plant cells, but centrioles are made from microtubules, and microtubules are found in plant cells. Okay. The mesh under the plasma membrane is made up of what cytoskeleton element? And the answer is microfilaments. It's the cortex. Microfilaments. And lastly, the mesh under the nucleus is made from what cytoskeleton element? That's called the nuclear lamina. And that's made from intermediate filaments. Okay. So micro for the cortex under the plasma membrane, and then intermediate for the lamina under the nuclear envelope. Next topic. We're almost there near the end. This topic is diffusion. So molecules naturally have thermal energy, okay? And that's just a result of movement. And the movement of particles into an available space, that's what we call diffusion. So while the individual movement of any one molecule may be random, like the the movement of a population of molecules can be directional. Okay, so now here's the core principle of diffusion. The molecules move down their concentration gradient. Okay, molecules move down their concentration gradient from areas of high concentration of that molecule to areas of low concentration of that molecule. Okay. And this applies to all solutes, and when that solute is considered to be water, that process of diffusion is just called osmosis. So now let's look at this drawing. Both sides begin with the same amount of water, okay? And let's consider this membrane to be non-permeable to the solute. Okay, so this membrane cannot allow the solute to move left and right across it. Okay, so the question here is, will the solute diffuse down its concentration gradient from right to left? And the answer is no, right? And that's because the membrane is impermeable to the solute. Okay, but the membrane is permeable to water, right? So if you think about it, there's ultimately more water on the left and there's less water on the right. And you can think of it this way. Like on the right-hand side, you have more solute molecules, so that leaves less space for water. And on the left side, you have less solute, so there's more space for water. And so because you have more water molecules on the left than, say, on the right, generally speaking, water will flow from the left to the right, and that is called osmosis. It's essentially equilibrating the concentration of the solute, because here the concentration of the solute is now equilibrated, okay? And that's because the water has gone to the right via osmosis and helped equilibrate the concentration of the solute. Okay. Clear the ink. So now, if the U-tube membrane was permeable to both water and solute, what would the final U-tube look like? So if this was if the membrane was permeable to both water and the solute. So the answer is that you would start off with a U-tube like this, right? And there was equal water on both sides, but you had way more solute to the right and less solute to the left. If the membrane was permeable to both, then what you would see is that the water level would stay the same, but the solute, because now it can flow in between the membrane, would just be normalized between the left and the right. If I added a lot of solute B, which is the second solute, different from the first, to the left side of the tube, okay? But the membrane was impermeable to solute B, okay? So B could not flow to the left and right. But say that solute A could. Would that affect solute A's diffusion from right to left or left to right or whatever way? And the answer is no. Okay, it's just a core principle to keep in mind that the diffusion of any one solute is always dependent on its own concentration gradient. So, for example, solute A was more concentrated to the right, so it will flow to the left. It does this independent of whether there is a second solute B that has a higher concentration on the left. It's not part of the calculation, so to speak. Like solute A will flow down its concentration gradient, and solute B will move down its concentration gradient if it's able to. given the permeability of the membrane. Now, in the right U-tube, which was, once the in the original example, what you had was a membrane impermeable to the solute, but water was able to move, and it created an equal concentration. So does water stop diffusing left and right even after this equilibrium is reached? And the answer is no. It doesn't stop moving left or right. It's just there's no net movement of water in any one direction. But it's still a dynamic state where there's still movement of water left and right. All right, on to tonicity, which is our last topic, and then we'll do some comparisons, wrap up with the summary, and then mention some details about our live class where we cover the full chapter with additional questions on every topic and live Q&A if you'd like to join for your own benefit. So, tonicity. A hypertonic solution has a lot of solute, okay? Hypertonic solutions have a lot of solute. Hypotonic solutions have less solute. You just have to get used to this terminology of hypertonic and hypotonic. And it's going to be either high solute or low solute. Water generally flows from which to which? It generally flows, or not generally, water will flow from an area that's from a hypo to a hypertonic area, generally speaking. Okay, and that will be in an attempt to equilibrate the solute concentration. So water will flow from somewhere where there's less solute to where there's more solute in order to help kind of dilute out the area that has more solute and equilibrate the concentrations. Okay, so now if a solution is hypotonic, that means that there is less solute outside, but there's a lot of solute inside. What will happen? Well, water is going to flow in, and it's going to puff up. For example, in the context of a red blood cell, it's going to puff up the red blood cell and it's going to cause it to lyse. That's why it's dangerous to create a hypotonic environment within blood cells, because then your red blood cells will just lyse and you won't be able to deliver oxygen to your tissues, and that can be a very fatal state, medically speaking. Now, in a hypertonic solution, there's a lot of solute outside and less solute inside. So water flows out of the red blood cell in this case, and it causes the cell to shrivel and die. So that is also a bad situation to be in, where you have a red blood cell in a high solute concentration environment, a hypertonic environment. Now, in an isotonic solution, the concentration of solute will be equivalent both inside and outside, and hence water will flow both in and out of the red blood cell, and you have a kind of happy isotonic state where the red blood cell is not puffing and lysing or shriveling up and dying. Okay. What do you call plant cells that are in hypotonic solutions? So remember, in hypotonic, the plant cell, if it's hypotonic, there's less solute outside, there's more solute inside, so water would flow in. And you call a plant cell in a hypotonic solution target. If you inject water into someone's artery, what would you expect to happen to the red blood cells in that portion of the artery? So you have your red blood cell, and now you've injected all this water into that artery, which is going to make the environment hypotonic, so there's going to be less solute outside. There'll be more solute inside. It'll be more concentrated inside, and then water is going to flow in, and then it's going to cause the red blood cell to puff up and lyse. A pond water paramecium lives in a hypotonic environment. Okay. Would you expect its contractile vacuole to be active? Okay. So there's your paramecium. It's in a hypotonic environment. That means there's less solute outside. There's more solute inside. So water is going to flow where there's more solute. So it's going to try and come and enter the paramecium. So this contractile vacuole, you would expect it to be active because it will be actively trying to pump out all this water that's flowing into itself because it's in a hypotonic environment. So the water would be pumped out of the cell. All right, that's your last topic. Let's go ahead and do a quick comparison of eukaryotic and prokaryotic cells. It's a very common area for questions and where a lot of students will make mistakes. So I did want to end doing a comparison of prokaryotic and eukaryotic cells and just highlight the common pitfalls. So number one, prokaryotes do not have membrane-bound organelles. That means no nucleus and no membrane-bound organelles, like the mitochondria, or ER, or GOLGI, etc. Prokaryotes still store their genetic material. It's just in an area called the nucleoid, and they still produce energy, even though they don't have mitochondria, with, for example, glycolysis. Number two, prokaryotes are unicellular, and they'll store their DNA in circular chromosomes, and they have 70S ribosomes. These eukaryotes will have 80S ribosomes. So prokaryotes have circular DNA as their main storage of genetic material, and eukaryotes, their main storage of genetic material is their linear chromosomes, but they still have circular DNA in their mitochondria and chloroplasts. All right, so what's common? What's common is that both will have ribosomes because they have to produce proteins, both have plasma membranes and enclose the cytoplasm, both have DNA just in different forms, and both have cytoplasm. Okay. Just don't forget that both have ribosomes. Sometimes people seem to forget that simple fact that prokaryotes and eukaryotes both have ribosomes. Secondly, let's go ahead and end with a final comparison of animal and plant cells. I think the biggest mistake students make is thinking that plant cells have centrioles or centrosomes. They do not. But plant cells still have microtubules, so remember that. Cell walls, chloroplasts, and central vacuoles are present in plant cells but absent in animal cells. And while plant cells have chloroplasts, that does not mean they don't have mitochondria. They do. Okay, plant cells do have mitochondria just like animal cells. Don't forget that. They still produce energy through oxidative phosphorylation. Plant cells and animal cells both have ribosomes because they have to make proteins, and they both have plasma membranes and Golgi and nucleus. What's specific to animal cells, though, are centrioles and centrosomes and flagella. Congratulations on making it to the end. We're going to do a rapid-fire summary of all of the topics. First, we covered the cell. Robert Hooke coined the term cell in 1865, And we talked about cell structure and organization. Cells can be organized into tissues, which are organized into organs, which are then organized into organ systems, which can come together to form the organism. We then covered cell theory. We covered how Schleiden claimed that all plants are made of cells, how Schwann claimed that all animals are made of cells, and how Virchow popularized the concept that all cells come from pre-existing cells. We covered the basic components of the prokaryote, which is a plasma membrane, a cell wall, a capsule with fimbriae extensions, and how the DNA is stored in circular chromosomes in the nucleoid region. We then covered eukaryotes, which also have a plasma membrane, but they store their DNA in linear chromosomes and a membrane-bound organelle called the nucleus. We went into more detail on the nucleus, which is a double membrane-bound organelle that is perforated with nuclear pores that have nuclear pore complexes. The nucleolus produces rRNA and is involved in ribosomal subunit assembly. Its structure is supported by the nuclear lamina, remember, which is made up of intermediate filaments. We covered the ribosome, which produce proteins. Prokaryotes have 70S ribosomes, while eukaryotes have 80S ribosomes. They can be ER-bound or cytosolic. We then covered the ER. The rough ER is studied with ribosomes and involved with the synthesis of secretory and membrane proteins and also membrane. Smooth ER is not studied with ribosomes and is involved with calcium ion storage, lipid synthesis, carbohydrate synthesis, and drug/poison detoxification. We covered the Golgi, which was discovered by Camillo Golgi in 1898. It modifies proteins via glycosylation, sulfation, and phosphorylation. We then covered the lysosome, which are vesicles that contain enzymes that are involved in the digestion of food and autophagy. Vacuoles, they're selective in what they permit to enter, and plant cells have a large central vacuole that stores potassium and chloride. We then talked about the mitochondria, which is the site of cellular respiration, and then we covered 3 different types of plastids. Chromoplasts that store dye, chloroplasts that conduct photosynthesis, and leucoplasts that are non-pigmented and store various macromolecules. We went into more detail on chloroplasts, which are the site of photosynthesis. We then covered the cytoskeleton. You have microtubules, which are hollow tubes made of tubulin dimers that are involved in the transport of vesicles/organelles, in chromosome movement, and the general support and structure of the cell. Then we covered microfilaments, which are these thin solid rods made of actin subunits that are involved in muscle contraction and support of cell structure via forming the cell cortex right under the plasma membrane. Lastly, we covered intermediate filaments, which are intermediate in size and form from various protein subunits like keratin. And the nuclear lamina, remember, is made up of intermediate filaments. We then covered diffusion, which is the movement of particles from areas of high concentration to areas of low concentration, and osmosis is the movement of water from areas of high free water concentration to areas of low free water concentration. We wrapped up with tonicity and discussed how red blood cells in hypotonic solutions puff up and lyse, while red blood cells in hypertonic solutions shrivel up and die. And then we finished up with 2 comparisons between eukaryotes and prokaryotes, and between animal and plant cells. So that's it. Congratulations on making it to the end. My name is Lutfi Hawk. I was your instructor today. I went to Phillips Exeter Academy, did my bachelor's at Cornell University, and currently I'm a PhD candidate at the University of Pennsylvania. I've done research internships at Stanford Medical School in California, at Mayo Clinic in Minnesota, and the National Institutes of Health near Washington, D.C. I'd love to share whatever I've learned, and if you found this helpful, come join us on our weekly live classes where we'll cover the full chapter with additional new questions on every topic, and you'll be able to see me in person, interact with me, and ask me whatever questions you have during the lecture and during the live Q&A at the end. If you have any corrections, suggestions, or comments, please comment down below. Go ahead and subscribe to our YouTube channel, CBSE IB Academy. Join the family for future updates and details, and I hope you found this helpful.